All right. So, welcome to the next 2 hours. Someone put into the chat if they cannot see my doctor. Brayton has something to say to me. Let me know if there's an issue seeing the PowerPoint or hearing my voice.
Sorry, Dr. Arbo, I didn't mean to send that.
Sorry, I didn't quite understand what you said. All right, I'm going to keep going unless I hear otherwise. So, you've got me for 2 hours today. And the next thing you have from me is in 2 weeks. That's asynchronous, and I will post those later today, so those will be done. And then you'll see me. I think I have some other asynchronous, but the next synchronous will be in about a month. So today we're going to talk about lipids to start off, and I already kind of hammer homed how important water is. Well, lipids are important because of water, because they separate from water and they cause this structure. And so while you are here to learn about human medicine, this would be true pretty much everywhere, we're quite certain. So on an alien world, they will likely have lipids in water or something like that, because the hydrophobic hydrophilic separation causes order, and you need order to create life. So more than an anatomy of just humans, I guess. All right. I'm not going to go over the objectives as I've mentioned. I will point out that there's this one objective that has a huge number of slides, the chemical structures. That's just because of how it had to be done in PowerPoint. I didn't want to have one massive slide with everything, so I phase it in. So this is a rare case where the number isn't really indicative of how important that is, okay? You're going to hear about some things you've kind of heard about in the lay press, so the omega fats and inflammation, and so we're kind of building the foundation. But we'll get into more nuanced stuff you haven't heard of, like Alzheimer's and cholesterol tie-ins, things like that. So on the right, top right here, didn't you mean to send that? I don't know what you're talking about, Brayton. Text me in the chat so I know what you're talking about if you're trying to convey something. Okay, so in the top right here, I have a saturated fat, right? When you see a V-shaped squiggly line, that is a carbon chain with just a bunch of hydrogens on it. And I think something that a lot of kind of, it's a little ivory towerish, but passes over students, is that when a fat is saturated, it inherently has more energy. So as you add oxygens to this fat, as it gets more oxidized, you can get energy out of it, okay? And so we'll talk about why we store energy as fats, right? They're energy dense, that sort of thing. So when we're looking at a big take home that students will often come up, actually, I guess it'll be come up to me this year. So normally students will come up throughout the semester, and a lot of the questions are kind of well targeted at, well, in my life, how does this relate? What can I do to get in shape? What kind of patient am I going to have? And so a lot of it comes down to metabolism and diet, right? And so here's a rough breakdown of how we spend our calories, right? So 60% is your resting metabolic rate. That is your heart beating, your brain thinking, you being warm-blooded, sleeping, you're burning that. And so this kind of can't be overstated that, like, we burn most of our energy just as maintenance, okay? So if you have, and especially for this lecture, but for all lectures, I'll be, usually if I don't say anything, I'll be talking about kind of the 2/3 of clinical patients you'll see. So in the Western US example, that'll be a obese type 2 diabetic maybe, okay? So I'll be talking about things about the issue of having too much weight. How do you lose it? There's obviously, you'll have classes, lectures about bulimia and anorexia. That's a different issue. But so if you're trying to lose weight, burning it by running is very hard to do. You clearly can do that, but like if you look at how much extra value meal has in it, and then think about how many miles you have to run, most of the all the data basically shows that the best long-term way to have a healthy weight if you're overweight is to change your lifestyle and lower the number of calories coming in, right? So I'm going to talk to you about biohacking and like how we can kind of use these biochemical pathways to on the edges do stuff, but it really is largely calories in, calories out, okay? CICO. So you'll hear a lot of things against that, and I'll teach you the things against that, meaning how can you do something that's not just calories in, calories out, but it really does boil down to that. And the reason that is, so this is calories in and calories out determine what your weight will be, right? The reason it is that way is really thermodynamically, that if you're burning more calories than you take in, then you have to lose weight. But the kind of reason people don't think it's calories in, calories out is because the calories out, the amount that you expend in energy, can change without you realizing it, so they don't count it as calories out. So for example, if you crash diet, you will lose weight, but you will lower your resting metabolic rate. And so this will come up in the Biggest Loser examples where they tracked everyone. And so if you lower your resting metabolic rate, that means how many calories can you eat in a given day if you're not doing anything. If that goes down, it's harder to keep losing weight or to keep weight off, because you go back to your, well, what do I normally eat? And now you're eating more. And so you'll rebound up in weight. Okay, so you have your resting metabolic rate, then you have the thermic effect of food, and then you have physical activity. Another thing I want to, let me move this. And I don't think I show you this. Shoot, that was a quiz question, but what is 1 calorie? 1 calorie is the energy required to raise 1 milliliter or 1 cc, cubic centimeter of water 1 degree Celsius, okay? And this is one of those examples where you can scale up to, okay, we have to keep ourselves warm-blooded, so how much does 1 calorie actually do? 1 milliliter is not that much, it's about that 1 cubic centimeter. So 1 calorie, scientific calorie, which is a little c, can raise that volume of water 1 degree Celsius. We clearly have to spend a lot more than that, but we talk about, getting something on the order of 2,000 calories a day. Why is that? That is because the food industry understandably didn't want large numbers. And so they're talking about, when you look at a food label, they're talking about kilocalories, which is 1,000 scientific calories. And the way you know that that's what they're talking about is they have a big C on the label, okay? So if you see a bar that has 25 calories, well, there'll be more than that. Let's say 100 calories on the food label, big C, that is 100,000 scientific calories, meaning you could raise 100,000 milliliters of water 1 degree, which makes more sense. All right. Okay. So it looks like we're good on the chat. So the breakdown of where calories come from, now I'm talking about the big C food calories. Carbohydrates have 4 calories per gram, proteins have 4, fat have 9, and alcohol has 7. And this is why 40 years ago, there was this big push to reduce your fat intake. It totally was understandable that, okay, well, if you're going to eat 50 grams of food, if you eat 50 grams of fat, you're eating more calories. There's starting to be an obesity problem. Let's get rid of the fat. And then we had all these things that had added sugars in them. So it's pretty clear now that fat is not bad. And if you were going to eat the same number of calories, well, anyway, in the Western diet now, people would probably do better if they ate more fat, definitely less carbs, less sugar, and we'll talk about all of that. And again, it gets at kind of the calories in, calories out thing, and that fat is satiating, meaning it fills you up. So you don't, if you eat fat, you actually don't end up consuming the same amount of calories in. Whereas if you eat carbs, you end up consuming a lot of calories in because it doesn't fill you up. All right. So I am going to try to slow down in the second half of this talk. In the first half, the part I'm about to hit, I'm purposefully not, and that's because we're just going through nomenclature. So I could have just said, this is assigned reading, but I figured you'd rather me just once through it to kind of hear what's important and what things are. So we've already talked about this is an acyl chain, so carbons with just hydrogens. You're going to hear carboxylic acid. This is a carboxylic acid with, I'm talking about on the bottom left, with the R chain coming off, and R means some carbon group. So you have a carbonyl, and it's COOH for the carboxylic acid. Whereas an ester has a carbonyl, a C double bond, but then it's going off to an oxygen in another R chain, another carbon. And we'll see these in fats. All right, so go ahead to poll everywhere. Let me make sure this is active, and click on the structure in this lipid that you think makes up most of our adipose tissue, our fat stores. Okay, 3, 2, 1. Very good. That is the right answer. This is triacylglycerol. Let me take yours away so you don't have to change your answers. So look at these structures, and we're going to walk through what they're called, and then at the end we'll see why this one is used. This is I understand why you would pick that, but this is the one and we'll talk about it. All right, so that's going to be annoying. I don't know if you guys can see this, but okay, I guess that's better. All right, so here's our first acyl chain, and this is called, here's this fat part, right? The up and down kind of mountains, and then the carboxylic acid. So this is called a fatty acid, fat carboxylic acid, okay? And if you have these, they can kind of spontaneously form this shell, this sphere, and this is all based on thermodynamic energetics, so it doesn't take, you don't have to like use ATPs or anything to do this. If you create these compounds that I'm showing you, they naturally form certain structures, okay? We'll talk about these getting oxidized. You'll hear about eicosanoids, and I'm just throwing this out there because I'm not covering this, but Dr. LaFontaine, another biochemist, will talk to you about autocoid signaling. So fats can be used in signaling, a whole nother thing that I won't get into. With inflammation, I will talk to you about the omega-3 and omega-6 fatty acids for the colorblind people here. The omega-3 is green because it's good, and the omega-6 is bad because it is bad. It causes more inflammation. Omega-3 reduces inflammation, and we'll talk about aspirin and ibuprofen related to that with COX inhibitors. So we'll get into that. Here's the next one, and this chain, this 3-acyl chain on the left is a triacyl, and then the backbone is glycerol. I'm showing it here, and that is called triacylglycerol, TAG. So this is can be stored, a lot of energy can be stored as fats, and we'll talk about the vesicles that this goes in. If you look at someone pre and post weight loss, so someone that was definitely morbidly obese or obese and loses weight pre-weight loss, in the left here is their amount of water, so we're a lot of water. Then they have in pink, purple, and in green protein. And then I won't, yellow bone, that doesn't really change. And then post weight loss, basically almost all the change comes from fat. It is true that your protein goes down, and I'll talk to you about biohacking there. The big take home is that if you crash diet, especially if you're doing strenuous exercise when you're not eating, then you will burn protein because you need instant energy and fat takes longer to mobilize. But if you do, I'm a big proponent of intermittent fasting. I'll talk to you about the science of that as we go on. It does look like all things seem to burn that protein a little bit, like maybe 20% or something. And if you do intermittent fasting, that seems to protect your protein a little bit more. So you burn more fat than protein. Okay. Here is again that glycerol, same as above, and then there's a phosphate on this one, and so this is a glycerol phospholipid, and this forms most of your membranes in your cells. Then here is a sphingosine chain that I've highlighted with another lipid added, and so this is called a sphingolipid. And here's a steroid, and those go on to form the sex hormones, estrogen, testosterone, cholesterol, and you'll have a whole other lectures. Again, you don't need to know structures from me. I'm not going to give you a structure, but you do need to know what each of these does, right? So like it's used in the membrane versus lipid rafts versus storage for fats. Okay, and so I'll give you 10 seconds just to think here. Do you see similarities in these and differences? There's a few ways you could do this. But, oh, actually, I don't think I'm hiding this from you. If I organize them this way and I circle these are all hydrophilic moieties, right? And so these compounds are amphipathic. That means they have a hydrophilic and hydrophobic section. And so if I draw a line here, you can see that all of this is hydrophobic, okay, on the left side, and all of the right side is hydrophilic. And the one outlier here is triacylglycerol, because that glycerol backbone comes back and all the oxygens are used to bind acyl chains. There's no hydrophobic group, and this is what allows it to pack and very well next to each other. It's all hydrophobic, and this is our fat stores. Okay, all of these would have problems packing because the hydrophilic end wants to be near something hydrophilic, and you just want to get it away from water and store energy. Okay. All right, any questions on that? How are we doing? So, I just told you that fat's not as bad as people thought. You will measure triglycerides in the serum, and that being too high is bad. So a normal level is less than 150 milligrams per deciliter. Borderline is 150 to 200, high is 200 to 500, and above 500 is very bad. And you'll see this increase of triacylglycerides with decreasing HDL, high density lipoprotein, which is good cholesterol, and increasing LDL, low density lipoprotein, which is bad cholesterol. These are the, buzz terms for these. HDL is bad cholesterol, LDL is good cholesterol. And that goes along with insulin resistance and metabolic risk factors. If you have not seen, normally I have a show of hands here, if you have not seen the plots that I'm showing here on the right, these are forest plots. If you see these, it's a good summary of scientific literature, because each line here is a scientific study. And so, this is the way science works. You do a study, it has to be replicated to prove it. And so this is looking at many studies. Here's an example of looking at medium chain triglycerides, MCTs, and weight loss. So switching to an MCT diet is useful in certain disease states. The most clear example would be like in epilepsy. You can change the way that what energy source the body's using, and so you can reduce seizures if you are on a MCT diet for certain cases. So, So in an MCT diet, you have this increased energy expenditure and weight loss, and it's a slight example, but here's the forest plot showing that the black diamond is the average over all of them. So you see this is looking at the mean difference, and so you have a weight loss of a little bit across these studies. And this is the change in waist circumference. Again, they're all beneficial slightly. All right, this is just a review slide. Whoops. Okay. So we're going to go through, again, a little bit of nomenclature. I don't need you to know it all. I'm going to point out the things you do need to know. So saturated, we've already talked about. Hydrogen's on the whole thing. Monounsaturated is you have a carbon that does not just have hydrogens. And then polyunsaturated, I have in green here, those are the good ones. We'll talk about those more. And saturated fats are bad, right? They're red. And there's actually been legislation to reduce these synthetic versions of saturated fats in processed food. So that it's very clear that will help society as a whole in reduce health costs and that sort of thing. So the fats can be branched or linear. They can be cis or trans. Cis is where the carbon bond kind of comes back on itself because of a double bond. Trans is in the opposite direction. They can be odd or even carbons, and they have these names. I only really will point out like 3 or 4 trivial common names that I want you to learn, because patients will talk to you about them and you'll hear a lot about them. And then I don't need you to know the systemic name. It does kind of very obviously tell you where there are double bonds, 9, 12, 15th carbon. The octa, deca means 18. Octa is 8, deca is 10. And then the omega nomenclature I do want you to know, and I'll talk to you about that in the next slide. So that's again telling you where these double bonds are. Okay, so here are saturated fatty acids with no double bonds, and the carbon, the scientific name is very obvious. The deca is the 10, so you see tetra 4, this is a 14 carbon, hexadecanoic acid 16, octadecanoic acid 18, icosanoic acid 20. And then if you think of the amino acids we've already talked about, this slide and the next slide can kind of toggle back and forth that these are the acids, right? Again, I think students get confused there because these have the hydrogen to give up, right? In the next slide, we are having like tetradecanoate, which has lost the hydrogen. So this is a little annoying on Panopto, but I'm going to toggle back and forth for you to see these. So, right, so look at these words, tetradecanoate or hexadecanoate versus ic acid, okay? And that's the same nomenclature as like glutamate or aspartate, right? Aspartic acid. Anyway, not a big deal there, but that's what that means. All right, so now this slide has the nomenclature that I want you to know. So there's numbering for the scientific numbering that starts at the business end. What do I mean by the business end? Where a chemical moiety could attack, right? So this could get esterified right here. There's a partial positive charge in the bottom right on that carbon because the oxygens are pulling it away, and so something can attack. So that's the first carbon, second, third, fourth, fifth. So in the scientific nomenclature, the number 1 starts at that carbon that can be attacked. But I want you to understand that, but you're not going to see that a lot. I'm not going to test you on that. The Greek lettering you will see sometimes, but that starts 1 carbon away from that. So the alpha is at the number 2 scientific carbon, alpha, beta, gamma, delta, epsilon. The nomenclature you'll see the most commonly, which is kind of the least scientific, is the omega numbering, and that starts from the other end. So on the left side here, this is an 18 carbon fat. Omega 1 is the 18th carbon there. So then you go 1, 2, 3. This would be an omega 3 fatty acid because the first double bond is at the 3rd carbon. Okay, so an omega 6 fatty acid will have that double bond start here at the 6th. Okay, so here are the common names I'd want you to learn, okay? So oleic acid and linoleic acid are 18 carbons. Oleic has 1 double bond, linoleic has 2 double bonds. And so you can see that linoleic is an omega 6 because if you start at the 18th, omega 1, 2, 3, 4, 5, 6, the 6th carbon is the first one to have in a double bond, okay? Alpha linolenic acid is also 18 carbons, but it's an omega 3 fatty acid. This is a better one, starting at the 18 for omega 1, 2, 3. That first double bond is here at the omega 3, okay? And then arachidonic acid is 20 carbons, but that's also an omega 6, omega 1, 2, 3, 4, 5, 6. The first double bond's at the 6, okay? So here are the 3 big ones to know in the bottom 3, linoleic, linolenic, arachidonic. I'm going to show you on the next slide. These are called PUFAs, polyunsaturated fatty acids. And PUFAs are generally good. Now, compared to saturated fats, right? So here is a memorize and a board testable, and there should be an action figure guy here because this is very actionable. So this is kind of the take-home slide for omega 3s versus omega 6s. So from the diet, you can get linoleic and linolenic acid. Now, we can't make the omega 3s and omega 6 fatty acids anymore. We need to get them in our diet. We've lost the molecular machinery to make them, okay? If you consume both linoleic and linolenic acid at the same time, this fatty acid desaturase, like one enzyme, can work on both of them. And so the key there is if you consume more linoleic acid, the enzyme is occupied and can't convert the linolenic acid. And so you end up kind of over swamping it, okay? And so this is why fish fats can be very good because the fat in fish is past this step, okay? So you can get eicosapentaenoic acid, EPA, and docosahexaenoic acid, DHA. You'll just normally see those as EPA and DHA. You should know both of those. Those are good fats because these are omega 3 fatty acids. These are in green that reduce inflammation, Okay, so the right side here is reduced inflammation. If you go to the left, linoleic acid, it goes on to become arachidonic acid. That is in red. That is bad because that goes on being worked on by COX cyclooxygenases to become eicosanoids, which cause inflammation. Okay, so very easily on the left bad, on the right good, and it's because that enzyme is working on both of them that you can overpower them. In the Western diet, we have about like 10 times too much linoleic or omega-6 versus omega-3s. So the take home here would be if you, as a biohack, if you were going to eat the same amount of food through, like let's say over a month, if you routinely had too much linoleic acid compared to linolenic, that would be bad. Whereas if you kind of toggled days of having, okay, here's my linoleic day, and then you had a just linolenic day, the linolenic day would go through and create some more anti-inflammatory lipids. Okay, so on the right here, I don't need you to know the bottom part here. See this clock with an FYI? What I do want you to know right here at the top part is that here's the arachidonic acid, and let me, where did that go? There we go. Let me record or draw this for you. So arachidonic acid, right, which is right here, which is bad. It goes on, to form prostaglandins, and I don't know how to change this color, but this is the color I want right now because it's red, because it causes inflammation. Okay, and now I do want to change the color. Dang it. I don't know how to change the color. But what is good is aspirin and ibuprofen because they block this, right? They block that step. They can inhibit cyclooxygenase, COX inhibitors. Okay. Let me look at the chat. Why does the numbering start from the end? The numbering starts from each end, so I don't know the answer to that. Like, I can't explain that. It's just for the omegas, you're caring about the where the double bond is, because we can't make that double bond, and so it is a lower number if you start from the other end to say omega-3 versus omega-6. Inflammation of what? So the inflammation pathways in general. So in a lot of disease states, you'll see we have too much inflammation, and so you want to hamper that down. You can cause tissue damage. Inflammation can cause pain, and this is minutia that I'll get into later. Massive inflammation is bad. Like if you look at a protein assay chip or an mRNA chip to see like in your biome what's happening at low levels, small amounts of inflammation can be good. So exercise, for example, causes small amounts of inflammation, but generally when you'll hear inflammation, it's at a level that's bad. Okay. So what can you do to kind of improve this omega-6, omega-3 ratio? So historically, in an evolutionary sense, we consumed about a 1-to-1 ratio in Western diets. It's now at 10-to-1, so 10 times as much omega-6 as omega-3, which we already talked about as inflammatory. Now, it's not as actionable as I said, because if you look here, this is on the graph, on the x-axis is the amount of omega-3, and on the y-axis is the relative strength of some beneficial effect. And the take home here is, this is obviously from a textbook that has kind of simplified, a lot of studies. In the light shading here, the light tan is how much you get from a dietary dose, and in the darker to the right is if you supplement with this. And the take home here is that you see these upward trends, and where do things plateau? So antiarrhythmia, heart rate lowering, blood pressure lowering, and antithrombosis, all of those seem to kind of have been plateaued or leveled off before you reach the dietary dose, meaning we consume enough omega-3 to get most of that benefit. The big outlier here is triacylglycerol lowering, right? So I showed you that having too much TAG in your blood was bad. Well, you are not getting enough omega-3s in your diet to kind of max out the benefit of lowering the TAGs. So if a patient that has high triacylglycerol, if you get them on a diet where they up their omega-3 compared to omega-6 from where they were, that should help that. Okay. And so we already did this one. Moving on. Okay, so here's just a fun slide for you guys, and I'll give you the data afterwards. Choose all the seafood below. I'm going to clear. I cleared it in case it was not cleared, that you have eaten in the last year, and then I'm going to show you which ones are the healthiest and why. And I'm going to give you 60 seconds so I can grab some coffee when you do that. All right. And you've got, I'll give you 10 more seconds. You can choose more than one. Click all of them that you've eaten. 5, 4, 3, 2, 1. Okay, so the top ones are all the good ones. I think they kind of don't taste great, unfortunately. Anchovy, sardines, herring, mackerel, rainbow trout I like. Salmon, a lot of people like salmon, that's great. Halibut and crab. And then tuna's not good. Tilapia, I'll show you why in a second. All right. So if I were a doctor, I would print this off and have it in my office to show patients. I love this chart. This is showing on the x-axis increasing levels of omega-3 fatty acids, so the DHA and the EPA that we just talked about, and so the right side is good. And on the y-axis, going down is increasing levels of mercury, so down is bad, right? You don't want mercury. So the top right is the best section. And so you see those things I labeled: anchovy, herring, sardines. Rainbow trout and crab are my best. Salmon I like, smoked but not cooked. You know, to each their own. If you know a really good cooked salmon recipe, I'd love to have it, but no one's convinced me yet. Whoops. I thought I had another thing here. Guess not. Let me just pop out real quick to make sure. I guess I'll get to that later. Okay. Let's start talking about why certain places use these fats and the properties they have. If you are hydrated, if you have no oxygens and you just have hydrogens coming off your alkyl chain, you can pack very tightly to each other. So here is steric acid, it's 18 carbons, and here's oleic acid, which is also 18 carbons. The difference is that oleic acid has a double bond, and it can be kinked. So it can't hydrogen bond, it can't pack and have energetic forces holding it together. And so because of that, it is liquid at room temperature, or its melting point is 14 degrees Celsius. Whereas steric acid, same length, its melting point is 70 degrees Celsius, and so it's solid at room temperature. And so when you start seeing, fast food places or things that use Crisco, they use things that can sit on a shelf and not rot, which are these solid fats, okay? And so commercially, it makes sense for them to do that, but it turns out for our health, that's just not as ideal, okay? So here we're going down to more and more flexible, okay? Cholesterol is, it is a fat in that it is this alkyl chain, but it's a very, very unique one in that it obviously has all these ring structures. But so cholesterol is very rigid, and I think I've talked to you maybe already, but I'll mention it again in this lecture a lot, that cholesterol rigidifies your membrane and your cellular membrane. So it'll be in these things called lipid rafts, which kind of aggregate proteins to do something at the cell surface. So when you hear cholesterol, think rigid for the membrane. So then the saturated fats can be solid at room temperature because it can pack. DHA has a lot of double bonds, so it can twist up like this, and so it's very flexible. All right. So cholesterol can, just so you can kind of see it here, it can pack into the lipid membrane with that OH group, the one hydrophilic thing pointing out, and then the rest is this hydrophobic thing packed in. And cholesterol, you'll have lectures or podcasts from me later on about this, and maybe in year 2 actually. Cholesterol becomes progesterone, pregnenolone, cortisone, aldosterone, and then that becomes testosterone, estradiol, and cortisol. So cortisol is a stress hormone, and it also has the sex hormones. So it's a starting point for all of those. So what else about cholesterol? Well, it is greatly increased in our brain. If you look at the percent body mass, it only makes up 2% of our body mass, but if you look in the brain, it makes up 20%. And that's because our neurons have these myelin sheaths, which can be thought of as insulation, like for a wire, you need to insulate the signal. And the myelin sheath is a fatty membrane that's enriched in cholesterol. Okay. That's all there. Okay. So we already talked about how we're basically big bags of water. And now I'm having a lecture on lipids and how important they are. Well, how are the lipids moving around? What happens when you eat a Big Mac? How does that get around in your liquid environment? Well, you need to pass it and store it as energy, use it in membranes. Long story short, you end up encapsulating these to shuttle them around in your liquid environment, okay? So there are these things called lipoproteins that have different sizes, okay? And so HDL, that good cholesterol, is high density lipoprotein, okay? This is showing the relative diameter. Then low density lipoprotein, bad cholesterol. Then there's VLDL, very low density lipoprotein, and then chylomicrons are the biggest to transport large amounts. Okay. So, you've heard that cholesterol is bad, right? But it turns out it's not, so you guys are too young, but a long time ago, I don't know, maybe 20 years ago, they were like, just eat egg whites. Maybe that's 30 years ago now. Don't eat the egg yolk because it has too much cholesterol. But actually consuming cholesterol is not bad. So look at these bottom 2 graphs. On the bottom left, you have your cholesterol intake going up, and on the y-axis you have your LDL cholesterol, your bad cholesterol. So so that we just showed you that. Sorry, let me go back up. This low density lipoprotein, LDL cholesterol, how much of this is there with cholesterol? Because that's bad. Whereas the HDL, the high density lipoprotein with cholesterol, is not bad. But as you increase the amount that you consume of cholesterol, your LDL cholesterol does not go up, okay? So this is flat, okay? So this going up, is that bad? This is bad going up, but it's not changing. Whereas in this one, the red box on the bottom, if you look at your plasma cholesterol, okay, going up, so this is what we've said is bad, plasma cholesterol, your deaths go up, okay? So it is bad to actually have extra cholesterol in your body, but you're not getting it via consuming just raw cholesterol, okay? And then I also wanted to make the point that people kind of talk about this. It sounds like it's more of an issue for men, but it is not. So the reason that kind of presents that way is because here we're looking at, in the top left corner, this is data for men, and then the top right is data for women, and we're looking at the lifetime risk related to cholesterol, okay? And so the black line is kind of low cholesterol, and then the high blue line is more cholesterol, right? So it's cut off on the left, but this is less than 200 for the black line, 200 to 400 for the yellow line, and then more than 240 for the blue line. And so you can see at age 60, there's a 5-fold increase for men between low cholesterol to high cholesterol measurements, right, in their blood. And then when they get to 80 years of old age, it's a 2-fold increase. Now the numbers are very high. The lifetime risk is around, I don't know which one you want to look at, but at older, it goes from 2.5, 0.25 to 0.55. My point is, if you look at the fold increase in women at 60, their difference between low and high is 8-fold. And at age 80, it's 2.2, about the same as men, fold increase. So while the women's lifetime risk is lower, if we're looking at 60, high cholesterol is only at 0.15, whereas high cholesterol for men is near 0.3, so almost double the amount. It's kind of for your patient, how much fold risk does it increase for them? So for women, it's still very important. For both of them, you want to lower that, right? Okay, and so here is talking about what happens when 1% of your diet from carbs is replaced with these different fats. Okay, so on the left, we're starting with kind of bad fats, and here's that. I had a slide, but I didn't really talk about it. The nomenclature of how many carbons, 12, colon, how many double bonds, 0. So these are all 0 double bonds, 12, 14, 16. Then you start getting double bonds with linoleic acid having 18 carbons and 2 double bonds. And the bars are white is total cholesterol, pink is LDL cholesterol, so bad, blue is HDL cholesterol, good. And so the take home here is that when you start getting into these green good ones that have double bonds, oleic and linoleic acid, the pink bar goes down, the LDL bad cholesterol goes down, and the blue bar, the HDL cholesterol, goes up. Okay, so this is talking about how that LDL and HDL cholesterol change, right? By taking different omega-3s, fatty acids versus saturated fatty acids, you're changing your HDL and LDL cholesterol, okay? Okay, so the fatty acids can be used for energy storage if they're esterified into triacylglycerol. They're used in membrane structures if made into phospholipids. Again, you'll hear about this autocoid signaling. And we've talked about cholesterol. It can go on to become vitamin D and bile salts too. So it's a precursor. Not only is cholesterol important for itself, but it also is an important precursor. All right, this I don't think is board testable, but it's important, so I cover it. So plastics are a version of fat, right? So when you dig up oil or pump it out, it's made from ferns and dinosaurs millions of years ago. And so it's high energy oil chains. But unfortunately, the plastics that are processed, a lot of them end up being endocrine disruptors, so they can mimic estrogen. And so BPA has been shown to do that. And if you kind of think about how can multiple plastics kind of be endocrine disruptors or mimic something like estrogen? I talked to you about cholesterol and testosterone and estrogen basically being alkyl chains fatty chains that are folded up into another structure. And so again, plastics are fatty chains, and the different fats or the different plastics have structures that can bind things evidently like some of our sex hormones. So here are phthalates, styrene, bisphenols, and estrogen, and just so you can see the structures and then where they're found. And so I've also given you the recycling code. So phthalates are number 3, styrene is 6, and bisphenols are mostly 7 and some number 3, so you can look and see what they are. And then they're health hazards. So phthalates influence PPARs, which you'll hear about, and produce insulin resistance. They can affect fetal development. Styrene can be inhaled indoors, so it's actually been good in COVID because people have been exercising, getting out more. They've been linked as a carcinogen for leukemia, and they're metabolized by P450. You'll hear about this enzyme again and again that metabolizes. We have this group of proteins that metabolize a lot of things, and so if you have something else that's hammering it, it could affect the metabolizing of other substances, right? Because that enzyme is being occupied. And then bisphenols are from the diet. They increase fat and decrease beta cell function. So we'll talk about diabetics with insulin and stuff like that. And so this is basically, I don't know if you guys have followed the plastic data coming out, but 93% of urine samples of 6-year-olds have bisphenol in them. And so basically plastics are everywhere. They've collected flowing air from the mountaintops in both the Rockies and in Europe, like so miles away from everything, and there are microplastics floating in the air there. So we can talk about that offline if you want. They've recently found organisms that can, we didn't think this would happen for a while, but organisms that can metabolize plastics. That's, in the lab right now, but that's a whole nother issue. How do we get rid of the plastics? If in another way to think about plastics they're an amazing carbon storage technology. So if you start metabolizing all the plastics, that will increase carbon dioxide, which is already an issue. So not tangentially related to health. Okay, so here's the summary. Yes.
Would you be able to look at the chat for a quick second? Sorry, it's kind of going.
Okay, thank you. Yeah, whoopsies. On slide 29, let me back up to slide 29. Sorry, I missed a bunch. Nope, that's not. Are my slide numbers different than yours? The graph shows that increased linoleic. Sorry, slide 30. Yeah. So oleic and linoleic acid are good. So this is, I think I know what you're saying. So I think that's comparing to this slide, right? So I wish that slide also had linolenic acid. The take home here is that saturated fats, and I'm not reading everything because I missed a lot of comments, but let me know if this answers it. Saturated fats are the worst, and then anything with double bonds is better. So both omega-6 and omega-3 can be better than that. And then within omega-3 and omega-6, omega-3 is better. Does that answer some of those questions? Definitely thought Mountain Air was fresher. Okay, are we good? Okay, I'm going to keep going. Thank you for, if I missed the chat, just tell me to go back and look. Okay, right. So here is the summary slide, saturated fats increase LDL. Then you go over, sorry, this slide was kind of talking about what I just got to. Then you have monosaturated fats and polyunsaturated fats. Polyunsaturated fats are the PUFAs, which are good, and they go down omega-6 and omega-3. Omega-3 are the better ones, but you can see that omega-6s also can decrease LDL, but omega-6s have these other beneficial properties. Okay, so here is, I think we're running short on time, but here is kind of all the fats rank ordered, and I'm showing you a few things here. First, we're looking at, in the blue numbers here on the left, I'm showing you the melting points, okay? The orange is bars of saturated fats, okay? And so you can see as you go up, the melting point lowers. So coconut oil is almost all saturated fat, right? This is yummy, yummy popcorn, although we haven't been to the movie theaters hopefully in a while. And its melting point is 25 Celsius, okay? So it's, that's very high. And whereas safflower or sorry, no, that I added one in chia seed, which is kind of the best one I have. Its melting point is negative 24 Celsius. So right, it's always liquid. And so these things that on the bottom left that don't go rancid, that last for a long time, are used, but these things on the top right are better. So on the right side, sorry, let me scroll down for the chat. Okay. So on the right side, sorry, there's a question, is linoleic acid in the diet good by itself? It is better than saturated fats, but it is not as good as the omega-6 fatty acids. Okay, so on the right side, we have in dark green, which is the first bar on the right, is monosaturated fats. Then in the light green, the next bar is polyunsaturated fats that are omega-6. And then in the yellow bar is polyunsaturated fats omega-3. So the far right yellow is the best line. And so I've highlighted two here, flaxseed oil and chia seed oil, because you can see they have these massive yellow bars of omega-3 fatty acids, okay? And salmon fat also has a lot of the omega-3 you see down there. So as an actionable item, I think these are hyperlinkable to probably Amazon. I wasn't paid by them, where you can buy them. Flaxseed oil is great. If we have time, no, we definitely won't have time. Okay, this is hyperlinkable. Maybe we have time after the next one. I gave Dr. Taffley and Arbo years ago and my son a taste test for these various oils with butter to see which tasted better. You can see the results. But long story short, chia seed oil tastes good with a lot of things. It's pricey though. Flaxseed oil is like a third the cost, or sorry, flaxseed oil tastes really good and is expensive. No, sorry, I was right. Chia seed oil tastes very good and is expensive. Flaxseed oil is like a third the cost, and I have a gallon of it, but unfortunately it goes rancid fairly quickly and it's kind of very nutty and hard to mix with stuff. So I'd pitch the chia seed oil. Okay. So here are the various diets. So we know the typical Mediterranean diet is good. And so you see a lot of fats that 38% fat, but you see the green bar where there's a lot of monounsaturated and polyunsaturated fat. If you compare that in the next row to the typical Western diet, you see that we have a lot more saturated fat. And then a low-fat diet is somewhere under 20% with a lot of carbs, and that's, while there's a nice picture of that, if you're eating a lot of fruits and veggies, I would say that's kind of frowned upon now more because of the carbohydrates we're getting aren't healthy carbs. Okay. If you replace 5% of the saturated saturated fats with these things, so we're replacing bad saturated fats with PUFAs, which are the best, MUFAs, monounsaturated fatty acids, carbs from whole grains, carbs from refined sugars, or trans fats, here's what happens to your change in CHD, cardiac heart disease. So PUFAs are the best. The change is minus 25%. Then monounsaturated 15%. Then carbs go down 9%. And then refined sugar and fat are bad. They increase it. Okay, and that's just kind of a summary. All right, any questions? Okay, I'm going honestly a little slow through the side. So what can membranes do? They can, once you have membranes, you can start creating different vesicles, which we do. You can have double membranes like the mitochondria, which we use for things. You can orient proteins in and out of a membrane for signaling. You can have double membranes, which I'm going to talk about. So there's an outer lipid and a double lipid membrane, or leaflet or layers, which we'll talk about. And you have to organize that. So it takes energy if we want to organize what lipids are outside and what lipids are inside. You have different proteins inside and out. You can interact with other cells as signaling, and you can start changing what's inside and outside. So Dr. Lafontaine will talk to you about the sodium-potassium pump. And as you're creating those ion concentrations, you're basically charging up a battery. Okay. This is just showing you the difference between various organisms. Here's us in the top left. We have that lipid bilayer. If you compare us to bacteria, bacteria have this cell wall that's made of peptidoglycans. And so we target that. I'm going to show you in the next slide. That's a good target because we don't have it. So a drug that targets that won't have a side effect for us. And so here is that cross-linker peptidoglycan layer, and basically we're targeting the cross-linking here, and that's how penicillin works, for example, by inhibiting that peptide cross-link. All right, so now we know lipids, now we know water, and now we know things that can get through and can't. So if you're small and you're not very charged, you can get through. So if you are a larger polar molecule, then you can't get through. Water shown here, I wouldn't really include water because it can get through maybe a little bit. Most of it's going through aquaporin and stuff like that. So basically larger and charged things don't get through. The lipid is, this is a cell membrane. There's an outer leaflet and an inner leaflet. And the outer leaflet, energetically, it can, one fat chain can move around left, right, forward, back, and rotate, but it cannot flip down, because energetically you'd have to take this hydrophilic head and bring it down through a hydrophilic head and bring it down through a hydrophobic area, right? So we have proteins that can let that flip, so we can organize what type of lipids on the outside versus the inside. And those are called flipases, flopases, and scramblases. And we'll go through those. So I drew it on the left here to remember if you start from the flipase. So a flipase takes a lipid and flips it to the inner leaflet, from the outside, from the exoplasm to the cytoplasm. And since you're creating order, since you're deciding where something goes, that takes energy. So it uses an ATP to do that. A flopase takes a lipid from the inner leaflet and flops it to the outer leaflet, from the exoplasm to the cytoplasm, or from the cytoplasm, sorry, outward to the exoplasm. That also takes an ATP. And then a scramblase just mixes up the lipids, and you're not creating order there. You're just kind of creating an environment where the lipid can go into a protein, not be exposed to a hydrophobic environment, and so it can go either way. And so a scramblase doesn't take energy. It requires calcium for that protein to be activated, but it gets rid of order. And we'll talk about a case where that's used. So the top left graph here is what you'll want to remember. So this, and I have a key down here. So SM stands for sphingomyelin. I made these up, I think. PC is phosphatidylcholine. And this is, the bar is showing you where that lipid, that type of lipid, remember I talked to you about the structure of sphingomyelin, where that is. Is it mainly on the outer leaflet or mainly on the inner leaflet? And so this blue bar is of total phospholipids. Obviously half is on the outside and half is on the inside, but for sphingomyelin, for example, most of it's on the outside, okay, the outer leaflet. So I've shown it, here's the cell membrane, and it's on the outside. Now for that to be the case, you know that something's working on it to put it there, right? I've told you if that would be a flipase or a flopase. I think I have a quiz question later. The main one that's very unique is phosphatidylserine, PS here, because you see it is 100% on the inside, okay? And so if your cell signals to create a scramblase for phosphatidylserine, then phosphatidylserine will start to be on the outside, and that can be used as a signal to kill that cell, because things from the outside can recognize that, right? And now say, okay, this shouldn't normally be here. This cell needs to be killed. So we've evolved for phosphatidylserine on the outside to be a death signal. This is just showing you that we start off as we're making our lipids and they're unorganized. So here, using that same key from before, PC, PE, PS are on the inside and outside in the endoplasmic reticulum. And then as you go to the Golgi, some of them start to get organized. And then in the plasma membrane, they're more organized, and I'll show that in another one, but they get organized as they go. This is another graphic showing that. If you want to work through how the lipids are kind of fusing, this will be obvious. This was a COVID year. If I'm a vesicle, okay, and let's say this, so I'm a vesicle going up, let's say this circle right here, okay? And so my tail, this part right here, is pointing outside of the vesicle. The tail is in the cytoplasm, okay? And let's say this top part is the membrane to the entire cell. As this vesicle comes up and fuses with the top part of the cell, it will go like this and fuse with the membrane, and see how this tail part, or what that was shown here, it's probably actually a head, but it was in the cytosol. Well, it's still going to be in the cytosol. So anyway, the point is the lipid layer that was in the cytosol as it fuses stays in the cytosol. That's pretty academic. I'm not going to test you on that. I don't know a clinical ramification, but that's how that's happening. Whoops. Okay. Well, actually, you'll be different than last year. So real quick, say which of the, oh no, we already did that one, sorry. Okay. We're down on time, so if you want to quiz yourself on this, I'll read it. Then hold on. Phosphatidylcholine PC is mainly present on the A or B, choose which one, side of the plasma membrane. So you're choosing one of these, extracellular or cytoplasmic. This is compatible with the fact that, and then answer C, D, or E axon it, flip base, flop base, or scramble base. So go ahead and close your eyes if you don't want to see it, and I will show you the answer. 3, 2, 1, I'm showing. Okay, so the answer for A and B is this one, and then the answer for C, D, and E is this one. And you can go back and look at that slide to be clear. Okay, we're off that slide. You can open your eyes. And here's the lipids showing that they do a lot of different things. You can secrete proteins out. You can engulf whole other things. Organisms engulf other organisms. So there's a lot of invagination. We'll talk about clathrin-coated pits and various things later on with these lipids. This is a big FYI slide. You don't need to know this, but I want you to see that what we just talked about, those lipid flipping things, if those go wrong, there are lipid flipping diseases, okay? And so here's just a list of some of them. You don't need to know these, just clinically relevant. We've already talked about how lipid rafts are formed with cholesterol. Okay. And I think this is one of the last few slides. Here's the tie-in for Alzheimer's. So again, you'll have a whole neurodegeneration lecture from me or from someone in next year, in year 2. And it's still not understood well, but the number one genetic link to Alzheimer's is this ApoE protein. And so we have 3 versions of it: ApoE E2, E3, and E4. And for the colorblind people, ApoE E4 is colored red here because it's bad and it makes you much more prone to have Alzheimer's. And ApoE E2 is good because it protects you against Alzheimer's. And ApoE E3 is kind of in the middle. And so if you look at the risk factors, the percent of population that has ApoE is 15%. If you look at E4, okay, the ApoE E4, the percent that has one allele that has E4, if you have one of them, you have a 3 to 4-fold increased chance of having Alzheimer's. If you have 2 E4s, if both of your ApoEs are E4, then you have a 10 to 30-fold increased chance of having Alzheimer's. So that's drastic. Okay, and percent of Alzheimer's patients that have an E4 are 40% compared to 15% of the population. And so how's this working? Well, ApoEs, we talked about these apolipoproteins. When they're bound to when they have cholesterol, they can decrease amyloid beta fibrils. And we talked about amyloid beta being bad, anyway, it's there after people have died from Alzheimer's. And if you don't have cholesterol in the ApoE, it increases amyloid beta fibers. And the take home is that the ApoE E2 has cholesterol and clears the amyloid beta fibers better than the ApoE E4. And now this is total non-scientific pontification that people have had, but of interest is that lower order primates, so primates that are not us, don't have these 3 ApoEs. They only have 1. And they also we can't use them as an animal model for Alzheimer's because they don't seem to get Alzheimer's. They don't get amyloid plaques. And so the kind of armchair professor anthropomorphizing no data type thing has been that what makes us unique in the animal kingdom. And so we have this amazing brain that uses a ton of energy, and maybe that churning is causing damage. And we have needed to evolve protective mechanisms such as ApoE E2. And so if you look at what genes have evolved or what class of genes have evolved in recent human history, there were only like 3 things that came up, and one of them was ApoE. And you have to kind of get into the grandparent effect for that to make sense, because Alzheimer's happens after you've had kids. But again, living as a grandparent could help you protect or nurture your grandchildren and get them through hard times, that sort of thing. The other ones are like facial recognition because we're social creatures and stuff. Okay. As another aside, we don't know about those amyloid plaques being so bad. It turns out that if you have 8 amyloid betas much, which normally the plaques have millions. Once you have 8, it might form a pore in the neuron and lyse the neuron, so maybe smaller amounts are bad. And this cholesterol can flopase, a flopase can flop cholesterol into ApoE. Don't worry about that. Okay. And that is it. Let me clear this. Go ahead and answer this, and we will come back at 10:15. Okay? Any questions? I'll try to answer the questions while you answer that. Is 4 an LDL? I don't know what that means. Sorry. So yeah. So remember, there's there are these vesicles that are fats that have proteins in them. ApoE4 is one of the proteins in that. Okay. So it's an apoprotein. Okay. Let's see how that was. Great. Okay. I will see you at the next recording. Yeah, I can disable that, or I'll try to do that. I tried to do it before, it didn't seem to work. So I think it changes the link, which could cause a problem. So. Recording, stop recording.